spiking neural networks. In 2001, he moved to Göttingen to join the Max-Planck-Institute for Dynamics and Self-Organization, and whose focuses on the research of physics in complex systems. And he started his own group as a group leader at this Max-Planck-Institute, and the name of this group characteristically is Neurophysics. And he is the first European who actually won the Mathematical Neuroscience Prize, which is very, which I want to emphasize. Since 2019, he's professor of physics at the University of Göttingen, and at the same time, the founder and also today one of the directors of the Göttingen Campus Institute of the Dynamics of Biological Networks, which is a joint venture between the Max-Planck Society and the University of Göttingen. And he's most known for models of cortical network development and evolution, and he is going to talk to us today as our last keynote speaker.
Yeah, thanks, Kerstin, for the kind introduction, and thanks to all the organizers for setting up this wonderful meeting. It's been a pleasure to be in your institute. It's a very impressive institute. Congratulations on the first 10 years, and let's make it even greater the next 10 years. I'm saying that because Kerstin, Markus, and Sigrid and us started an active collaboration that's applied the biomedically motivated research network on early impacts of Alzheimer's disease causing molecules. And I'm going to give a talk on spiking neuron circuits, cortical circuitry, information processing, and the use of that to understand early stages of AD when I'm coming back. So after we've done a year of work. But today I want to take you to another end of neuroscience, to the very fundamental question how complex, big and powerful brains like yours evolved, and whether the major transitions, the big inventions in neural processing that happened on that way are not also very important subjects for us to consider in theoretical neuroscience, and they can provide cues about how biological nervous systems are model information processing devices that can hardly be obtained by just trying to copy the human brain, the mouse brain, the fly brain, you name the NIH type of species. Actually, it's related to our project because one of the driving insights that got us to collaborate on this and got us the grant is that there's a widespread recognition that in AD models, rodent models are limited. AD spontaneously occurs in large and complex brains like in non-human primates or in carnivores. And so therefore there's no way around lifting the research to a more advanced model species like us. And so today is kind of the natural history background, how it came that these brains are kind of different and potentially more powerful. Okay, but before I do that, I often start these talks with a little bit of reflection on theoretical neuroscience, because I mean in theoretical and computational neuroscience we do different things, augment data analysis, try to get classification going and so on. That's all very important, but then one thing that's key to our study is to answer why questions. Yeah, so there are phenomena, and we want at the end of the day in biology understand why is this structure good for that function. And traditionally, physicists like me or people from computer science that try to answer these why questions try to suck an answer out of their little finger or try to import it from engineering. But biology, and many of you will be biologists, actually has a framework, kind of a theory for answering the why question, and that's the theory of evolution. The natural history and our understanding of how evolution shapes organisms, and the brain, just like all other complex organs, is a product of a long stretch of evolution. So that brings up questions that are normally not so frequently asked in theoretical neuroscience. So if you learned your 101 in evolution, there are qualitative fundamental questions about these processes. Whether the system that you are studying has evolved by gradual modification, stage by stage, or there are actually also processes in the natural history of animals that are sudden, all or nothing changes with a lot of stasis after that. And we need to understand how this punctuated versus gradualist evolution plays into the structures on our brain. Many people believe that the nervous system is a super conservative system to evolve, and there are probably fundamental reasons to that. Then for us as theorists, if you grab randomly a theory in computational neuroscience that wants to answer the why question, it will be an optimization theory with 99% probability. It's going to be an optimization theory, but then if you actually expose that to a biology audience, normally they say, well, nothing is optimal in biology, good enough, and so on. So that if one takes biological evolution seriously, then it's a big question mark whether we are actually in business in biology. I'm gonna self-critically address whether that works and try to convince you that there is actually optimization in brain evolution. And I mentioned in the beginning, so if we want to construct the brain as an information processing device, it has a technology history. The natural history of the brain is the history of information, living information processing technology. And if you think of any kind of technology, if you understand it, there are major inventions in that history, like for mobility, mobility by muscle power, mobility by combustion engines. Hopefully that's ending soon, and we realize finally that electrical motors are much more efficient and actually would enable us to survive on this planet. So, and these big transition points, I think if computational neuroscience has matured as a field, it should have something distinct to say about them. So that's my thesis. I'm gonna come back to that at the end. And so in that fashion, it's kind of the opposite of the current approach in much of the field to take the human brain and reverse engineer it with artificial systems that are superhuman in performance. I'm not sure that's going to give us too much insight about the human brain, or to pick any individual brain. So slicing the history into the major transitions is a worthwhile research strategy next to reverse engineering a given brain that you think is the most telling. So I'm going to talk about convergent evolution. That has a systematic reason, because convergent evolution is the phenomenon in biological evolution which is the clearest fingerprint of a process of functional adaptation and functional optimization. There are so there are hedgehogs, for instance. In Germany we have hedgehogs. It's good to have spikes. There are hedgehogs in Africa. They evolved completely independently. Being a hedgehog is a success model in living overground. There's a similar thing underground. So convergent evolution is success. If you find convergent evolution, these are success stories in the history of living technology. And again, that can happen in different ways, and convergent evolution with major innovations is probably where we should start with this. So if you consider the evolution of a neural circuit, circuit A prime, they are kind of different, but serve roughly the same function. That can happen gradually, or there could be kind of a fundamental transition in between where you switch from one network architecture to another. And if that is actually driven by such a technology spur, then you would recognize it in that there's another lineage that keeps the traditional circuit, the ancestral circuit, and does the same key innovation again. That's the thing that I'm hunting for, and there are a couple of these key innovations that are well understood. So the emergence of nerve cells and synapses is kind of the latest example. Some of you may have followed that in the last 2 years, evidence became very much stronger that nerve cells evolved 2 times independently in the history of life. But more common is centralized nervous systems. So many early nervous systems were not centralized. There is discrete centralization. That happened not only once, but several times independently in the history of animals. I think the invention of neocortex and say of the mammalian type of hippocampus are key innovations. Probably they happened only once, but clearly we know that there are key innovations in nervous system evolution, and some of them, the ones that should be most easy to read, happened independently several times over. So I'm going to briefly advertise that the other big thing that's happening, although not as many people are talking about it, next to deep neural networks and AI, is that evolutionary biology is going through a revolution. It started like 20 years ago, triggered by the big sequencing projects, and it's continuously expanding. And in that process, actually, in independent parts and distant parts of neuroscience, evidence for convergent neural circuit evolution has started to appear. So we had talks about vocal learning. Yeah, not very many vertebrates are vocal learners. Yeah, there are examples in primates, but we are pretty lonely as vocal learners among the primates. The other branch in vertebrates biology that is a great vocal learner are songbirds. And apparently there are close parallels in the circuitry in the songbird brain and in the human and primate brain that enable you to become a vocal learner. So that's probably not conserved. That's certainly not conserved from the common ancestor of us, dinosaurs, and songbirds. I mentioned neurons, centralized nervous system. We did a contribution to this kind of growing wave of recognition of convergent evolution in the nervous system by looking at the visual system, and that's the subject of my talk. So I said I want to kind of advertise that looking at evolution is great, and looking at this kind of science now is especially rewarding. So several things have happened since the Human Genome Project made large-scale sequencing so much greater. One is that our understanding of what the real phylogenetic relationships are has matured tremendously, because sequences are so much more rich and indisputable evidence for relatedness than morphology. And so one thing that everybody takes for granted now, but was found out only during that, is that rodents actually are very closely related to primates and humans. They're much closer than cats, sheep, and dogs to us. Then by molecular clock techniques, one can reconstruct when which lineage split happened. One fascinating thing is that all the major lineages of our model animals, of modern mammals, they separated way back in the Mesozoic, when basically all the ecomorphotypes were kind of a mouse. Yeah, and largely because of the big investments in another big country, China put hundreds of millions into paleontology. There is a fantastic database of fossils, and because computed CT became so more, micro CT became powerful, paleoneurology has turned into a really quantitative field. We're running in Germany a national platform for computational and evolutionary neuroscience, and one of our associates, Ornella Bertrand, published 3 years ago this science paper where they tracked size and morphology evolution of all the mammalian lineages that are present today, plus extinct sister groups over the past 150 million years, pinpointing where encephalization bursts happened, where actually the brain shrunk during evolution. So we get really the trajectory of brain evolution in quantitative detail because of the investment into paleontology and the advancement of structural imaging in fossils. Okay, so that's kind of the background why I think it makes a lot of sense now to look into paleoneurology, to evolutionary biology, to phylogenomics if you are a neuroscientist, because the structures that we are studying, we understand now and can tell much more certain than any time in the past. when these structures were actually invented and in what kind of animal they were invented. And that will give us part of the answer to this question of what is the purpose and how is it a functional adaptation. So the question that I'm looking is kind of one key riddle in cortical structure and function in the visual cortex. Is pretty simple, but I'm going to spend the entire talk on it to look at it from different angles, because quantification in depth is what we think in physics gives you answers that you cannot get any other way. So all of you learned in basic neuroscience that in the visual system, form vision relies on a system of edge detectors that are at the first stage of cortical processing. Hubel and Wiesel got the Nobel Prize for discovering these kinds of feature detectors in V1 for the first time, and they actually in their Nobel address stated that this is a wonderfully ordered structure. It would be a nightmare if the neurons would be randomly arranged that code for that extract different orientations. One of the first discoveries of single-cell resolution brain imaging, calcium imaging, by Clay Reid and his co-workers was that nightmare is true in rodents, in the mouse brain, in the rat brain, in the squirrel brain. Neurons that code for different orientations are next neighbors. There's a little bit of correlation if you are aficionado, but basically it's completely random. Hubel and Wiesel's nightmare. Of course, they used the classical model of visual cortical physiology, cat and rhesus. And there you have the highest degree of order that you can imagine. Yeah? So you go from cell to cell. There's a continuous progression of receptive field properties, with the exception of little singular points that many of you may know. So there are discrete points in the cortical tissue where all the orientations are in close proximity, and there's actually a jump. Yeah? But that's the minority. That's just points all over the place. There's continuous progression down to single cell resolution. And surprisingly, while kind of 20 or so species have been scanned now, there seems to be nothing in between. Now you could kind of try to mix it, make this more disorderly. That seems not to happen. It's an all or none transition. And Kerstin mentioned it. Yeah. Kerstin likes to ask the question, we don't know whether it's related to function, whether it's related to perception. Well, something must have mediated the transition from one to the other. It could be an epiphenomenon, but a few differences in these circuits are pretty obvious. So with the rodent typical salt and pepper arrangement, in every piece of the visual field, you have the entire repertoire of contour detectors. Whereas if you try to bunch the cells together in local groups, 100 or so micron in diameter, then it's easier to connect the ones that have to work together if you want to build a cortical amplifier or something. But that comes at the price that in every little piece of the visual field you are blind to the other orientations. Might be very hard to demonstrate that blindness psychophysically, but mathematically it's nearly unavoidable. So this is great for diversity. That's great for recurrent excitation, pattern completion type of optimization. That's hypothesis, but I think it's plausible hypothesis, and we should inch closer to see whether these advantages actually play out in the visual systems and the way these animals try to see. So what I want to present today is 3 chapters in how we approach this. The first one goes to the heart of this question of optimization. Yeah? So that it's either order or disorder is only one aspect of an all or none transition. The other question is there are many kinds of ordered arrangements. And so how these neuron groups are distributed over the visual field might still be different. And it's an open question whether you should view it as an optimized design or whether it's a good enough irregular design. So that's what I'm going to talk about in the first part and show you that there are real mathematical invariants of that circuitry that amazingly biology finds no way to break out of over tens of millions of years of evolution, which we still need to understand. Yeah? Then I'm going to look closer into primate evolution and focus on South America, the New World monkeys are a unique place to test the resistance and the adaptability of the visual system. You know that. So I can't use this pointer well because I'm colorblind like 99% of mammals. Primates, old world primates, reinvented color vision like 40 million years ago. With the exception of a few males here in the room, I'm belonging to them. New World primates have this weird systems where you have color vision in some animals but not the others, in females, not in males. But also examples, and I'm coming to that, got rid of color vision altogether. So if you think back at your neuroscience lecture, well, that raises the question of blobs. Yeah? So we teach our students there's color, form, and motion in vision, yeah? And you know what color is processing. I'm going to talk about that in this test case. And then at the end, I'm going to step back and address the question whether that's really, so how should we think about this all or nothing transitions? How can they be produced by modifying animals genetically, physiologically? And why and when did that happen? So let me start with the first thing. This part is concentrated on the ordered design, not on the disordered design. I'll come to the disordered design later. And the key players, so this is what I tend to call the form vision core circuit, is the complement of the connectome that gives every neuron its preferred orientation, and that is, according to the Hubel and Wiesel model, the set of efferents that either the neuron gets from the geniculate inputs, that's the scheme that's working in carnivores, or inputs from layer 4 to the upper cortical layers in primate cortex. But these neurons are densely interconnected. Kerstin showed this system of connectivity. Sigrid is one of the pioneers to demonstrate that it's formed by activity-dependent plasticity mechanisms. And so we know that these contour detectors don't work on their own, like the contour selective neurons in a deep neural network, but they are interconnected with far-away contour detectors that are preferentially wired if they have the same preference and even the same axial selectivity if you want. So if you want to model this, you need the minimum choice is to keep all the connections that give a neuron its afferent receptive field, plus the connections that make it work in a big network at the stage of V1. And so these things together, if an optimization theory holds water for that, have to be co-optimized. So of course, it's a hugely complex system with many billions of degrees of freedom. If you would go for every synapse, so what we do is to use a trick from theoretical physics to go to a reduced system of variables. One is a field-like variable, so if you can tell the preferred orientation and the selectivity at all the places, that is a field. I'll call it Z of X here. But you can't get rid directly of this system of interconnections. So the minimum system that we have to set up our optimization theories with is these maps plus the system of interconnection. Making the assumption that something is optimized means that the set of reasonably good states or finally optimal states reside in a much lower dimensional manifold in that state space, and the motion and the optimization on that submanifold is what we can capture with this reduced representation. And so in physics, there's a cool trick that you can import into visual neuroscience, not into auditory neuroscience, not into somatization, but I think there are generalizable lessons that we can learn from that, and this trick is called symmetries. Yeah? So I don't want to suck out of my little finger the optimization principle that your visual system is trying to realize, but I know that it has to deal with ensembles of images and movies, because that's the input, and any design has to work with an entire ensemble of images and movies. But images and movies have a lot of symmetries, because, so to start with, all of you can be at different distances, so there's a scale invariance to images that's unavoidable. Everybody can move to the left, to the right, to the back, and to the front, so there's translation invariance in the ensemble of images. It's also hard to tell the mirror image from the original. We had the problem that causes with learning to read and write. So there's and all of these symmetry transformations make up the Euclidean group E2. So whatever is the optimization principle, if there is one, it should be invariant under transformations of the inputs by the Euclidean group and by transformations of the visual cortical circuits under the Euclidean group. So that simplifies life a lot. Then you can start to kind of count and parameterize all the optimization principles that you can imagine. But in physics, we have the tendency to say, well, maybe it's even more symmetric than what you can reasonably assume. And so Euclidean symmetry means that you have to deal with any reasonable ensemble of images and movies. Then there's another additional symmetry that's used a lot in the field that uncouples the representation of cardinal and oblique orientations. And there is another little symmetry that I introduced actually 20 years ago that guarantees that detectors for all orientations are present. That's not trivial. You could have optimization schemes where you rather go for the cardinal orientations and ignore the oblique ones. And to a certain extent, that's actually happening in visual systems. But so having the highest symmetry class allows you to be sure that all detectors are there, that basically the oblique and the cardinal system are optimized on their own, and that you can treat, that you can deal with a realistic image ensemble. And surprisingly, mathematically, then the layouts, the joint layouts of horizontal connections and of the maps can be calculated with paper and pencil. Now, I'm not going to give you the talk where I go through this calculation, but trust me. So I publish it. It got accepted. I got a prize for it. So believe me, the calculation is correct, and it contains totally stupid physicsy type of neural circuit architectures. So if you imagine that there's the one best design to arrange orientation detectors in a little piece of visual cortex, the best design for all of V1 would be to take that little unit cell and copy it. Yeah? So you would end up with a crystalline arrangement. And certainly they are found in the state space of these optimization models. So if you wouldn't know all the solutions, you could conclude from this that because visual cortex circuits are so disordered, there's no unit that ever repeats. That's kind of evidence against optimization. Optimization, best unit circuit, and then you repeat that thing, like in barrel cortex. That's not happening in vision. Is it optimized? Well, what I discovered by this calculation is that there's a huge set of other optima that never repeat in space. And that's and that's also a phase of optimization models in which there's not one best design. That's another kind of basic biological concern against optimization theories. If something's optimized, there's the one best design, and then every individual brain should be the same if there's this one best design. Okay, math shows us that there are optimization principles that predict diversity, that predict a large degree of inter-individual variability. So these solutions, they come in, first of all, in manifolds, and I calculated the dimensionality is quite high dimensional tori, and on top of that you have a combinatorial number of these manifolds. So the state space is filled with ground states. They are not dense, but there are quite many of them. And so because they looked like the measurements that Sigrid and Tobias Bonhoeffer and Kerstin were doing when I was a graduate student, we thought of ways to quantitatively compare the predictions of this kind of the simplest complex system theory with real measurements. And these topological singularities, where neurons of all preferred orientations come together, give you a tool to do that without reconstructing the entire connectome. So that's again me stepping into my stupid Prussian physicists being, these are points. So if there are discrete things, what a physicist will do is to count them. How many are there? That's something that will distinguish different designs. They come in two kinds of flavors, left turning and right turning, depending on how you arrange the detectors around the center. And therefore, there are actually quite many statistics that you can derive from that. So there's the distance to the opposite type of singularity, and there's the distance to the closest distance to the same type of singularity, and the closest distance to the to any kind of singularity, and then there are holes in between where you don't have a singularity, and their size distribution. So that makes kind of a toolbox of quantifiers that are quite precisely accessible from data, but they don't require you to tell the entire connectome of that circuit. But if you know them, I mean, it's a good mental exercise. If you would know where all the singularities are and what their topological charges are, you can fill in the iso-orientation domains, you can start to color them and basically recover the entire layout just from the positions of those. So that's why that might be a useful starting point to compare theory and experiment in this system. And together with Matthias Gaschuben, our professor at Frankfurt University, I managed to, again with paper and pencil, calculate how many you have. And we have also expressions for all of these various distributions. Okay, so there's basically only symmetries going into this. What can be the result of such a calculation? There must be mathematical constants in it. It could be E, it could be square root of 2. It turned out to be the circle number pi. Yeah? So if you go to longer and longer, this is the range of horizontal axons. If you go to reasonably long horizontal axons, you converge to this mathematical invariant. So that's a theory that has a unique prediction, no parameters to tune, so it's bound to fail. We assembled a data set of like 200 brains at this time across the mammalian tree, basal primate, the galago, the model carnivore of the day, the ferret, and tree shoes, close relative to primate lineage. And well, the grand average in that data set was between 3 and 3.18. So the prediction is actually within this below 10% error bar. So I was hoping to see systematic differences between these species because they have quite different visual environments. But it seemed to be universal. Later we expanded it to include also cats, because these animals happen to have basically the same size, so you could imagine that there's some elementary. I'm going to come to that. But when we published this, then Kill Miller thought it's kind of one of the most precise measurements in systems neuroscience, and broke with the golden rule in computational neuroscience. With every equation that you put, you lose half of the audience. Yeah? So our technical papers are not read by anybody on this planet, but Ken Miller put the equation into the title of his comment. So that theory kind of worked much better than I could have hoped. I did it as a mathematical exercise to get started, but it seems to be the thing that explains biology across many species that have our kind of cortical architecture. And then I want to do another step and more critically test this. Yeah? So is this mathematical invariance really biological universality? And primate evolution is kind of the best way to test whether this prediction really can be broken. Because, I mean, many of you might know that most mammals are actually not like us. We are diurnal, but the majority of mammals is nocturnal for interesting reasons in evolution that I can go into at the end of this talk. But some of them break into the day niche. Yeah? So the basal primates, galagos and lemurs, are largely nocturnal and still have a tapetum. They didn't have color vision reinvented yet. in the pathway that leads to old world monkeys, apes, and eventually humans, there's a transition to diurnal lifestyle. The best evidence for that is that we all don't have a tapetum, so that was lost, and it was lost apparently only once in primate evolution. Then some of these monkeys rafted over from Africa into then an island continent, South America, and filled a lot of primate niches here, like 40 or so million years ago. And only one lineage, the old monkeys, reverted back to nocturnal lifestyle, losing also the blue cone. They are green cone monochromats. They are nocturnal visual specialists without a tapetum. So the old monkey is kind of the primate that made the wildest travel in terms of visual habitat. Its ancestors converted to diurnal lifestyle, then they invaded a new continent, filled many primate niches, and one of them, the nocturnal primate niche, was refilled. So if there is an evolutionary path with radical changes in visual environment that could shake these mathematical rules, it's in New World or old monkeys. So let me recap a few of the changes that happened. So the Galago is kind of primary nocturnal and lost the blue cone. The macaque, the primary primate model system in vision, it was chosen because it's like us, cone trichromat, and like us has a fovea. So that's also a discrete invention in primate visual evolution. And the old monkey kind of did most of the changes that you can imagine in visual system evolution. So after that story, and because I got a prize for mathematical invariance, maybe it's not a surprise that nevertheless, also the old monkeys don't break the rule. So macaque, old monkey, Galago, there's no evolution that we can think of, but these are species that are around for 55 million years, and that's a lot of time for mammalian evolution. In 6 million years, you can turn a cow into a whale. In 4 million years, you go from Lucy to us. So it's actually a question whether that invariance is inertia or whether it's optimization. Okay, no, I'm not going to comment on the short difference. But so the fundamental questions that it raises, this long persistence and persistence in spite of big changes in visual habitat, is whether maybe for brain evolution, these 55 million years are just a blink of an eye, and so that you cannot do much in 55 million years. Can we test this? And the color system, the blob system in V1 functional architecture proved to be a way to test it. So I solely looked at orientation columns so far and orientation detectors, but the very same neurons can be color sensitive more or less, can reside in CO blobs or in the outside. So V1 in primates is partitioned into regions that are CO rich, in which high rate firing neurons of reduced orientation activity are located, and the C in between, that's CO reduced, in which typically orientation activity is a bit higher. So there is a coupling between being a CO blob neuron and whether you're an orientation detector or not. So we set up a next level optimization theory where we try to optimize where do you put the CO blobs. If you want to minimize the impact on orientations and activity, you should move them to places where you're not compromising a well-clustered orientation domain, pushing towards the singularities. If you want to just cover the visual space uniformly with high active neuron clusters, then you should arrange these CO blobs in a hexagonal pattern. and anything in between. So this is again a theory that can be analytically solved. We introduced a correlation C star. That's actually a third order correlation. Now last evening we talked about third order correlations. That is a higher order correlation. And interestingly, if you push the parameters to really maximize orientations and activity and move the CO blobs out of the way of the orientation system, then there's an upper limit of this correlation. You cannot completely avoid inferring with the orientation system by moving CO blobs. They have to be someplace, and that's showing up in this optimal positioning limit. Now here you see this C star calculated from joint measurements of CO domains and orientation maps in the large ensemble of large-scale images in imaging data from V1. This is the theoretical limit. These are our 5 macaque measurements. They are all within the order of magnitude of the maximum achievable, and one of them actually nearly maximizes it. So in macaque, the positions of the CO blobs relative to pinwheels and domains are nearly optimized. So can this change? Well, if this is a color-associated system, the CO blobs, then it might change in the nocturnal species. And so in old monkey, both, and in the galago, so the primary nocturnal species and the secondary nocturnal species, this correlation is almost an order of magnitudes lower and is nearly indistinguishable. So evolutionary time in primate evolution is good enough, is enough time to move CO blobs to optimal positions and to some other positions back and forth with the change of visual environment. It's just the orientation system that is invariant. The CO blob system is very plastic. And so you can redesign V1 functional architecture, and it has been redesigned across the course of primate evolution. We are currently finally pushing to prepare this, and the reason that we haven't completed this story yet is here we understand what is optimized. But the question is whether the nocturnal primates optimize something else, what that other cost function is between which they are switching. So so then, having established this invariance when it comes to kind of the major stretch of primate evolution, the final point in the primate lineage to look at is the very beginning. So I told you initially that rodents are actually very close relatives to us. So perhaps there are transition stages, brains with a mixed architecture where you can see the act of transforming one cortical architecture into the other in a frozen or intermediate state. And that would be in the most basal lineages that have a environment and the ecomorphotype that's similar to the primate ancestors. Now, primate ancestors, like all ancestral mammals, are kind of 100-gram creatures like our friends from the lab, rodents, mice and rats. And so the living primate that is the best proxy of the ancestral ecomorphotype is the smallest primate there is, Microcebus, the mouse lemur. They are found in Madagascar and close relatives to galagos, but much smaller. So they are fitting into your hand. The data that you're going to see next is from 50-gram primates, the smallest primates there are. That's work done in collaboration with the group of Daniel Huber in Geneva, and the mouse lemur investigated are in the biggest mouse lemur colony in the world. That's a Paris research colony, and that was founded actually in order to support aging research and Alzheimer's research, because mouse lemurs are not only the smallest primates, they're also the primates that age the fastest. Having a lifespan of only 2 years in the wild, they become senile and develop dementia in 2 or 3 years, even faster than the marmoset. So that's why French research invested into establishing mouse lemur primatology, and we made neuroscience use of that. So there's two things that we looked at. One is this: how many singularities do they have, and it's another confirmation of this total invariance. We also measured the size of these domains, and that is the one thing that is not completely conserved in primate evolution, but only weakly so. So what you see here is the size or spacing between orientation columns, how many neurons are you grouping together, how small is the space, and many structures in the brain can be miniaturized. If you think of the insect brain, there is orders of magnitude of change in mushroom body, in the eyes, in the optical lobes. So there's no reason per se that nothing in brain circuitry cannot be miniaturized, but orientation domains can hardly be miniaturized. So there is a little trend to make smaller columns in the smallest primate brains. But basically, the columns in Microcebus that are the smallest in mammals so far are within the range of macaque. So there's no scaling up of column size, and there's no scaling down of column size when you go to small animals. And note, here we have the little orientation cluster in mice. There is a little tendency to have more similar neurons in close proximity, like 50 micron correlation radius. I see no way that this is a scaled down primate, although they are called supra primates. There seems to, that's another data point that confirms that there's really all or none transition between the rodent visual system and the primate visual system. Oops, let me turn on. So, and of course, so I promised you there are very many statistics we looked at. We didn't go by a single number, and all of them are falling on top of the other primate and carnivore species, and on top of our simple, relatively simple model. So to wrap that part up, primate orientation domains seem to be incompressible. You cannot scale them down in size substantially. Mouse orientation clusters are really something different. One could imagine that when you go to a smaller and smaller primate, you approach the salt and pepper locally clustered architecture. That's not the case. This invariance is a unique feature of the form vision circuit. If you go for color, there is evolutionary transformation. I didn't mention it. It's more classical. Also, eye dominance, how the two eyes are brought together, is different in different primate species. So V1 functional architecture in primates is plastic on evolutionary timescales, but the orientation system is not. That's a genuine puzzling finding that it's so invariant. And in the CO system, there must be at least 2 repositioning events in modern primate evolution. So in the last 5 minutes, how much time is left? Is that okay? You're still with me? Yeah? Okay, I'm gonna look into the how and why, and when. Maybe I'm gonna skip the how. That's the part with the modeling and the simulation, and maybe so we are going to be around next week if you want to know how you should redesign the synaptic plasticity rules in the brain to switch a monkey into a mouse or the other way around. Come ask me, I'm going to show you our simulations of that. I'm going to go back to the natural history. So if there's an all or none transition, there's a question of whether in the history of life there's a plausible place for when that might have happened, or an idea for the circumstances that might have triggered or allowed switching from one kind of cortical architecture to the other. And there is, so actually as part of the work of the International Panel on Climate Change, the history of the past 90 million years was closely investigated in the first report period in search for past examples of rapid climate change. So there's still the question of whether over relatively short periods of time our planet could switch to a totally different pattern of local climates. And in order to study this, actually paleontologists and climate natural historians engaged in big campaigns to re-examine the fossil record, to collect new cores and fossils to see whether major climatic changes that are always associated with mass extinctions, smaller or bigger mass extinctions, happened in the recent past. And one such event is actually 55 million years ago, 10 million years after the dinosaurs got extinct, and we mammals basically inherited the world. And there's, so in this, at the boundary between the Paleocene, the direct period that followed the extinction of the dinosaurs, and the Eocene, when modern mammals appeared, kind of your hallmark modern mammals, horses, primates, appear. Notably, so for the biomedical researcher, rodents are not really in this modern group. The first rodents appear already in the Paleocene and seem to smoothly transition between Paleocene early forms and the modern rodents that we see. Primates are very different. They appear all or none over a horizon that's maybe 50,000 years long. Really, that's really a flick. And what happens between the older primate stem group fossils and the first true primates is several things in parallel. One thing is really visual. So that's bringing the eyes to the front. You are well aware that most vertebrates have the eyes to the side, and that's for a good reason. You want everybody's food. You want to see your predator even if it's coming from the back. You have a price to pay to bring all the two visual organs to the front like we do. And there are theories for that. We are very social, and we can have somebody else cover our back, and monkeys do that. That happened in this all or nothing transition, together with a massive expansion in the brain, mostly at the back, because the tectum that was previously exposed in stem primate ancestral brain reconstructions is now covered. So there is a simultaneous repositioning of the eyes to the front and a massive investment into the brain. And that's thought to be related to another climate change episode, because at the transition between the Paleocene and the Eocene, this is a world that is already 12 degrees higher than today, a hothouse world, and then there's an additional 3 to 5 degree temperature peak that only lasts 100,000 years or so from rise to end. But in this time, actually the entire planet was covered with tropical forest, rainforest, dry forest, but you have tropical forest in Antarctica at that time. So that is the kind of planet where it makes sense to invent the primate lifestyle and the primate ecomorphotype. And the leading hypothesis, in my opinion, to make sense of this is what Sussman's so-called angiosperm coevolution hypothesis. It's not just about the primate being under evolutionary pressure to become smarter. It is that a huge energy resource becomes available by this massive expansion of forests. So by teaming up with fruit-bearing trees, primates serve the ecological purpose to disperse the seeds of the angiosperm trees, and in exchange get these sugar balls that are more and more designed to feed our energy-hungry brains. So there's kind of a runaway circle, according to Sussman's hypothesis, that if you specialize for the fruits, you have an energy source to power a much bigger brain, and a much bigger brain allows you to travel all the fruiting trees that can feed you at different times of the year, and navigate that space and be a good disperser. So tapping into a new source of energy might have enabled this encephalization burst. And becoming a visual specialist, actually, that's still nocturnal. Okay, so in the part that I skipped, I would have showed you that in the state space of optimized neural circuit, there's a phase transition between the salt and pepper phase and this pinwheels and domains phase without an intermediate stage. So by tuning a few physiological variables, you can switch between one and the other. If you want to know more, come talk to me while Sigrid and me are here. So a gradual modification of cellular rules can lead to all or none switch. And the rich paleobiology of primate evolution that we have now points that there's a likely place for this transition to happen, and it's at the time where the niche for primates actually happened, and brains are costly devices. It's the most energy-consuming kind of tissue in our body, and that expansion needs to be powered, and it could be powered exactly at that time in Earth history. So the picture that I try to point to you is actually not the thing that we convey in our textbook neuroscience introductions. The stuff that I talk to you, orientation columns, orientation detectors, are neuroscience 101. But actually, it seems more like they are the most derived state. Although you would think, if you have a good neuroscience training, that that's a basic universal feature. But when neocortex was invented, it seems that we went from a stage where actually the forebrain mantle was still more organized like the hippocampus, and there's no topology and no receptive fields at all if we take the turtle cortex as the model of that. And then with the invention of neocortex and the cortical column, there's not yet the invention of the functional column and the modern kind of clustering of features and activities. That only happened then after modern mammals and finally big primates emerged. So in this deep time perspective, the things that seem to be really simple and the one-of-one of neuroscience are highly derived properties of the brain that went through through several stages of fundamental changes in technology. Okay, so current projects to better understand this is, of course, to find what are good candidates for these optimization principles. I didn't have to suck them out of my little finger, but eventually we want to answer what this kind of architecture is really excelling at. That's what we are currently doing. Then you cannot talk about evolution without addressing genetic control. So how the architecture of the area-wide architecture of upper layer networks is genetically constrained or instructed is very poorly understood. And if that's a useful thing, one would expect that even if learning mechanisms can set up this architecture, there might be a genetic backup system, and we are trying to work out with how little genes and transcription factors and signaling molecules you could encode that in primate genes. We are still not able to do neuroscience with PhD theses that last 50,000 years or 100,000 years. So in order to recapitulate that eventually there has to be another way, but genetic modification. But with optogenetic interfaces, you can build circuits of living neurons that you can reconfigure on the fly, and that's one Julian, who visited Kerstin just a few weeks ago and presented this approach at your bioinformatics meeting, is working on a platform, so you can, in a tabletop experiment in an afternoon, go through a neural circuit transition that's starting to mimic what might have happened in the course of primate evolution. And we are really putting a lot of effort into understanding what that kind of vision is that our ancestors did, because it's in the middle of the night. Actually, so mouse lemurs, we are working together with the German Primate Center group that's studying mouse lemurs in Madagascar. They are active at night. They are only active at night if the moon is not out. They are active in the middle of the night when the moon is not out. That is a visual environment where your photoreceptors catch 3-5 photons per minute. It's really not the visual world that we are used to think in terms of vision, but we need to understand it better if we want to understand what drove that transition. And with that, I would like to thank the funders, our co-workers. That kind of doing theoretical neuroscience is only possible if you find really kind experimental neuroscientists like all of you that collect more data than would be needed for a typical paper, help you sort out what is a well-curated data set and wait till the end. And I'm very grateful that we have all of these fantastic co-workers like Kerstin and Sigrid. Thanks for your attention.
Thank you very much for this really interesting talk and this extreme nice summary. Okay, so are there questions?
Thank you so much. That was very inspiring. I was curious because in mice you have these genetic columns. They're loosely associated with the dominant, columns. Do you think this in mammals or in newer mammals, you have this genetic constraints of this genetic like sister cells that are forming these columns, and this is one of the driving forces of this process?
Yeah, thanks for the question. That's a very important point that you're raising. Yeah, so you're talking about the ontogenetic column, Pascoe-Rakic, and actually the stem cell community is the one that's put the most amount of work into understanding how you can build a primate-like brain. A lot of that work is in the stem cell niche, that more supragranular cells are produced. You have additional kinds of stem cells in the primate brain, it seems, than in rodent brains. Could it be another basis for the transition? I think that could be one mechanism. Yeah, I'm not sure I would favor it, but the stem cell account could go in two ways. There are two theories that one can build and test. One is that the migratory path of the excitatory neurons might be different in rodents than in primates. So there are better and better techniques to label all the sister neurons that come from one stem cell. And in mice, the evidence that I know from a group in IST, Austria, for instance, is that in the mouse, the clonal group disperses over like 100 micron or even 200 micron. So there are old papers from Rakic where these migrating cells are stacked one on top of the other. In the best mouse studies that I know, that's not the case. Yeah? So it could be a better ordering of the migration along radial glia that would make this clonal group stick together. And then if orientation preference is a cell fate that they inherit from the stem cells in the epithelium, then it would be a natural way to create salt and pepper and functional columns by migration of the neurons after the final decision. There's a second theory that could go along that way, and that if that was right, then there should be a proto map of preferred orientation of the functional columns in the epithelium that encloses the vesicle. Yeah? So that's where the stem cells come from. So certainly this epithelium has positional information, frontal, so AP-DV axis in gradients of signaling molecules and transcription factors. So if there would be a genetic circuit to read out positional information in the epithelium and then inherit that to all the system neurons, that would be a prerequisite for that migration theory for the transition to work. Okay, but that's an epithelium, and we have also biophysics of epithelia. Now, epithelial cells are active, mechanically active cells that can actually change their topology, and whether they preserve the topology while being a contractive epithelium or not depends on the tension of ectomyosin at the tight junction that connects them. So the other theory would be that in the first stage, the neurons read, the epithelial cells read out the position information, make an orientation map, and then if it would be the cortical tension in the epithelium that enables them to mix before they generate the cell fate. But actually, I don't believe in the cell fate theory, yeah? But I believe that working that out mathematically would be great fun. So if a student comes and wants to do that, that would, I think it's an idea that should be worked out and tested. But a simple test would be to really do a good labeling experiment of the daughter cells and see whether in primates they stay in a line and in rodents they are dispersing. That's, I think there's good evidence for that. So, okay, so two answers. Yeah? So there's a lot of variability when it comes to the actual dimensions. So I showed you that on average you cannot scale change the size of columns from big to small. But if you look at an individual animal, then there are parts of so we did that in cat visual cortex very extensively together with Sigrid. There are parts where you have big columns, parts where you have small columns in the same animal. And Sigrid kept such a good record of her cat experiments that we would know which animals are siblings and which are distant relatives or not related at all. So in one of our first joint papers, we measured this variability in the size of domains and tested whether it has a heritable component, whether siblings are closer to one another, and how much that heritable component is compared to the total variability. Long story short is we could demonstrate that there's a heritable component, so there's genetic control over this system. At the same time, the largest amount of variance is idiosyncratic. You cannot predict it neither from where the neuron is in V1 nor from the sibling. So I think there's strong evidence that there's genetic control. Whether it's hardwired, I think it's doubtful because of that big variability. So that said, I wouldn't question your other point that there might be genetic control, because genetic control can work in different ways. Yeah, so in the kind of cell fate determination way. So you're born a horizontal neuron with the last division from the stem cell, and that would be kind of a forward determination. But we know that there is signaling between cells and the markers and the gene regulatory circuit that they turn on are determined by interactions with neighboring cells. So I think that is a very interesting direction to further push the thing, because axons are not only electrical cables to transport action potentials and cause exocytosis. There is forward axonal transport of signaling molecules. There's translocation of signaling and transcription factor molecules across synapses and backward transport. So the kind of genetic control that can work with few genes and with a lot of variability would be to make the cell fate decision in every neuron not be determined by the stem cell, but by the interaction with other genetic circuits through the same cables that transmit electrical activity. So.
Hi, friend. So, I was curious actually about the parameter that control the phase transition. What's the parameter that control the phase transition? Yeah.
Okay, so this gives me permission to give another talk. So first, let me hammer home our basic credo, namely that you cannot really understand the system of modules without the system of horizontal connections. So what I'm showing you here is to prime you on the answer to this question with two simulations. One generates a salt and pepper rodent-like state, and the other one generates a primate-like ordered system. What you see here in the corner is the horizontal projection pattern of the neuron labeled by a dot, and that's also here. So here I exposed the horizontal circuit that's optimized, and in the rest you see the projection of the most upper layer neurons, and there you see actually this is a 3D simulation across the entire cortical depth. So does this work? And actually they're starting at similar initial conditions, and in this ordering phase the neurons order in the Z direction because they're closely interacting through the anatomical column, and the connectivity is arranging itself into long-range patchy connections that preferentially link the same neurons. In the what's with my second? In the salt and pepper phase, basically the rules for refinement of connectivity are very similar, yeah? But because the neurons are not grouping in cells, then the connectivity becomes much more fine-grained and is specifically linking neurons that are in this interspersed configuration. So here is the, okay, no, I won't show that. So here's the phase diagram of what is basically going on. The control parameters that we tested here, potential control parameters, are the functional wiring specificity. So Kerstin mentioned that there's always a little bit of discussion. Is like-to-like connectivity only to neurons within plus minus 10 degrees, or is it basically to everybody with a certain preference for the same orientation? So down here would be disregarding totally the other preference. Down here, up here is total specificity. The red region, this red corpus, is the disordered phase, and it basically has flat walls. You know? So even with a little bit of specificity, it works well. If you take it away, salt and pepper still forms. What is important is the excitation inhibition ratio and the size of the excitation inhibition footprint. So if you are dominated by inhibition above a certain level, you know, like a blanket of inhibition, Rafa Juste at some time talked about a blanket of inhibition. Of course, there's specificity in the inhibitory subcircuits in cortex, but the basket cells basically innervate everybody pretty densely. So with a blanket of inhibition and the footprint that is within a certain tolerance, you get salt and pepper for free. And by shifting the excitation inhibition ratio into a bit more pattern completion, excitatory currents, the cortical amplifier, and having the excitation inhibition footprint such that a Mexican head kind of thing emerges, then you go to this column design. Yeah? And so that's a direct first order phase transition.
Thank you.
Thank you very much. Is there another last question? Otherwise, I will finish the discussion and ask, why should we study South American marsupials, and do we expect a phase transition?
Yeah, okay, so why am I here, yeah? Okay, I'm going to indoctrinate Kerstin for the next 68 hours until you can wake her 3 o'clock in the morning and answer this question. But so you should be proud of opossums because there is there's actually a long-standing hypothesis of convergent evolution of one lineage of opossum and early primates. The woolly possums, Calyromis, actually are the most encephalized opossums and have a niche that's very much like the mouse lemur or the small lemurs of Africa. And in anthropology, it's a long-standing hypothesis that they are a model of primate evolution. So that's the one lineage in which there must have been kind of an encephalization trend or an encephalization burst. When you look into their face, there are eyes to the front, and they actually, they don't have a post-orbital bar, but a little post-orbital thing in order to stabilize the eye. So that would be a lineage. Then the other smoking gun or interesting place is the split between what Lea Krupitzer and increasingly also me believe to be the most primitive and ancestral-like marsupial, which is Monodelphis. So genetics and a lot of anatomy and a lot of belief has it that Monodelphis is the least derived opossum. But the group that is most closely related to it is the Marmosa. And Marmosa actually have almost reached the encephalization quotient of Calyromis. So that would be really amazing, if this kind of transition between salt and pepper and calms that in eutherians happens at the ordinal level. So characters that change in evolution at such a high taxonomic level are believed to be very hard to decode, because it's a shared feature of all members of the order that invaded so many different niches. How can you find a correlation with a lifestyle or ecology that supports that feature? So salt and pepper versus pinwheel is kind of an ordinal trait, and most evolutionary biologists would give up trying to solve what is the driver. But in marsupials, it might actually happen below the family level. And okay, I can go on about marsupials. So that's the hidden reason why we study Alzheimer with Kerstin in cats, so that we can start to do research on marsupials in South America. Thank you for the invitation.
Thank you, Fritz. So that was a nice closing talk, and now we can also close our symposium, and then I would like to ask Claudio and Tarciso. First I want to thank Claudio for this very great organization, because I think he did the major part of it. We can agree to that. And I also would like to thank all the helping hands, the students in the background, and the technicians, Gabriel, Haisa, representing all the others who also helped. And I would like to actually ask to come everybody to the front who was involved in the helping, so that we can make a photo. And then and clap with the hands. Raisa. Huh? No, you can. Saindo aqui. Vamos. You have to. Yeah, no? No? Nobody here any longer? And then we
I just would like to not thank you everybody who organized the symposium, but I also would like to ask you a round of applause to everybody who participates in the House Symposium with their posters, with their oral presentation, with their presence here. So thank you very much. It's you that make the symposium every year better. So a round of applause to all of you. Then I would like to invite Sergio Ruschi, who has the brilliant idea to create the first chess tournament of the House Symposium. I hope next year we will have the second chess tournament, and he will call the champions of yesterday evening. So, Sergio.
Boa noite, eu vou falar em português. A gente resolveu fazer esse ano esse evento, acho que foi bom. Eu queria agradecer a Cláudia por ter comprado a ideia assim, e apoiou. A gente fez um torneio, foram 19 inscritos, 14 que participaram, foram 9 homens, 5 mulheres, acho que foi bem representado, assim, 9 laboratórios representados, então assim, tivemos uma boa representação de todos os laboratórios. Esse ano a gente vai premiar só os primeiros colocados, então a gente fez 3 categorias. As categorias são campeã feminina, então eu vou falar do terceiro para o primeiro. Em terceiro lugar ficou Raíssa Breit. Em segundo lugar, representando o laboratório do professor Marcos, ficou Luna. E, em primeiro lugar, feminino, representando o laboratório do professor Tarcísio Velho, ficou Giovanna Arende. Giovanna está aí? Não, você. Agora, o campeão absoluto, em terceiro lugar, ficou o professor Marcos Costa, com 5 pontos em 7. Em segundo lugar, representando o laboratório do professor Siddhartha, ficou Caian Matos. E em primeiro lugar absoluto, fazendo 6,5 pontos em 7, Adriano Torte. A gente também fez a pontuação por laboratório, então os 2 melhores de cada laboratório pontuavam e assim gerou a classificação, a pontuação do laboratório. Em terceiro lugar ficou o laboratório do professor Adriano, que ele pontuou sozinho, mas foi o suficiente. Em segundo lugar, o laboratório do professor Draulio, com a pontuação de Nil e Marcelo Falck. Em primeiro lugar, com a pontuação de Luna e Marcos, o laboratório do professor Marcos foi o campeão. É isso, fica o convite para o próximo ano, todos participarem e os laboratórios terem mais representantes e a gente fazer um torneio ainda maior.
Pessoal, uma salva para o Sérgio por ter organizado isso. Muito bacana. As I will switch to English again, just for our invite to understand. So we also I would like to give a prize to the best oral presentation and to the best poster presentation for the PG Neuro, so our graduate program students. So just to be sure, only the students that are enrolled in the PG Neuro program were evaluated. And this year we have T-shirts for just a second. I put some of the T-shirts here. So the best oral presentation. So just to be sure, we have a committee, a very high-level committee that attribute points to certain given criteria. So we average these points for each one of the committee member, and then we find the winner. So, but I would like to say that I was very happy with the scientific quality of the oral presentations and the posters. It was very difficult to find a winner of this year. So it was very close. All the students were very close to each other, but we have to find one. So the best oral presentation goes to Flora. And the prize is a T-shirt from the Brain Institute. So here it is, Flora, congratulations. Valeu. And the best poster presentation goes to Gabriel. Valeu, Gabriel, parabéns, congratulations. And as a final thank you that I would like to, as a committee, would like to say, I know this is the last house symposium of our director, as a director, so I would like to thank you, Professor Kerstin and Professor Bernadette, for leading this institute forward. You are amazing doing the job in the last years. I don't believe that we will be where we are if you do not have your leadership. So I would like to thank Professor Kerstin and Professor Bernadette for being the head of this institute, the energy that moves us forward, and to keep the building running. So it's very important. So thank you very much. Thank you very much. So thank you very much. Professor Bernadette. We know that having this position is very difficult. Sometimes you skip part of the science that move us, but I believe it's your job is making every one of us happy to be working here in the Brain Institute. So thank you very much for the two of them. Gente, finalmente a gente conseguiu uma coisa que há vários anos a gente vem tentando, que é um desconto no Mahalila. Então, quem for levar o crachá até às 8 horas tem 20% de desconto no Mahalila para comidas e bebidas. E eu espero ver todos vocês lá para a gente celebrar esse final de ano, esse ano de 2025, que foi incrível para o Instituto. Então, acho que nada melhor da gente celebrar embaixo de uma árvore, tomando uma cervejinha artesanal, comendo uma moela com caçava, esqueci, macaxeira, e a gente possa conversar um pouquinho mais e, quem sabe, né, discutir outros assuntos de ciência. Hã? Vamos comer macaxeira e tomar cerveja. Gente, muito obrigado, uma ótima sexta-feira e um maravilhoso final de ano para todos vocês. Valeu!